The APOBEC3 family of cytidine deaminases in primates and humans has 7 members that act as host restriction factors against retroelement, retroviruses, and other DNA viruses that contain single-stranded DNA intermediates. The family includes 5 members that have been reported to inhibit HIV-1 in absence of its WIF protein. In absence of WIF, the APOBEC3 proteins can get incorporated in the budding virions along with viral RNA. When these virions infect new target cells, the APOBEC3 proteins can inhibit viral replication by 3 mechanisms. First, they can bind to viral RNA and directly inhibit reverse transcription in a deaminase-independent manner. Second, they can deaminate cytosines to uracils on the minus strand of cDNAs, causing G2A hypermutation and leading to non-functional proviruses. Third, they can cause hypermutation of the viral cDNA, leading to its degradation prior to the integration. However, lentiviruses such as HIV encode a protein called WIF, which antagonizes APOBEC3 proteins by causing their degradation. The protein of my interest, A3C, has been found in majority of studies to be weakly restrictive for HIV-1. Yet, it is highly expressed in CD4 positive T cells, can be encapsulated in HIV-1 virions, and is targeted by WIF for degradation, suggesting that it has a role in lentiviral restriction. Moreover, it has been reported that A3C has evolved under positive selection in primates in a manner that suggests that A3C has played a role in blocking viruses. A single nucleotide polymorphism in human A3C causes a serine at residue amino acid 188 to change to isoleucine. This polymorphic form was reported to restrict HIV-1 replication 5 to 10-fold more than the common human A3C form. This increased activity was due to the formation of a dimer. However, this polymorphism is specific to human A3C only, as the old world monkeys have only isoleucine and great apes like chimp and gorilla have serine at position 188. Interestingly, despite having a serine at position 188, chimpanzee and gorilla A3C are reported to be more efficient at restricting HIV-1 replication than human A3C. Chimp and gorilla A3C have achieved this increased activity through dimerization that is mediated by a unique amino acid, which is lysine at position 115. In the model shown here, the amino acid at position 115 needs to interact with arginine at position 44 of second molecule of A3C to form the dimer. Interestingly, rhesus A3C has amino acid isoleucine at position 188, but a different amino acid, methionine, at position 115, which is different from both human and chimpanzee A3C. We wondered whether these amino acid differences affected rhesus A3C antiviral activity, so we made 2 single mutants of rhesus A3C, changing amino acid 115 to either lysine or asparagine, and cloned them in mammalian expression vector pcDNA3. We used these constructs to do a single cycle infectivity assay to compare the restriction activities of different A3Cs against pseudotyped HIV-1 with no WIF protein. We observed that the human A3C S188I was active against HIV, but rhesus A3C was not, despite having the isoleucine at 188. Moreover, we found that changing single amino acid at position 115 does not increase the activity of rhesus A3C against HIV-1. We checked the expression level of our constructs to make sure that they are expressed at similar level. In addition, all the A3C tested encapsulated in virions at similar levels. Since amino acid 44 played a role in dimerization of at least chimp A3C, we analyzed the amino acid sequence alignment of A3Cs again and noticed that amino acid at position 44 and 45 of rhesus A3C were different from that of human or chimpanzee A3C. So we hypothesized that amino acid 44, 45, and 115 of rhesus A3C are inhibiting its dimerization. To test our hypothesis, we made a double mutant that has its amino acid 44 and 45 changed to arginine, and two triple mutants that have arginine at position 44 and 45 and either asparagine or lysine at position 115. We used this construct to do a single cycle infectivity assay. We found that all the mutants have around 2-fold more restrictive activity against HIV-1 than wild-type rhesus A3C. However, they were still not as restrictive as human A3C S188I. Again, the expression level of all constructs was similar and all of them encapsulated in virions at similar level. Since previous studies have reported the activity of A3C is correlated to its ability to form dimers, we purified wild-type and mutant rhesus A3C using SF9 insect cells and used size exclusion chromatography to determine their oligomeric state. As you can see, the triple mutant with asparagine at position 115 had two peaks indicating formation of a dimer. However, the monomer peak was still predominant. However, the wild-type rhesus A3C and other mutant showed only a single peak, indicating they were monomers. Since the N-triple mutant had increased dimerization, we determined whether this correlated with increased deamination activity as previously reported for human and chimp A3Cs. So we did an in vitro deamination assay. We first did a time course of deamination to determine the linear range of activity and then calculated the specific activity in that region. We found that rhesus A3 mutants that we have tested have approximately 2-fold more activity than wild-type rhesus A3C, indicating that changing amino acid at position 44 and 45 increases the activity of rhesus A3C. Although changing amino acid 44 and 45 to arginines in rhesus A3C increased its restrictive activity against HIV, it was still not as active as human A3C S188I, so we generated a model of an A3C dimer. From this model, we hypothesized that serine at position 144 creates a repulsive force for serine at amino acid 46 that can destabilize the dimer. Interestingly, both human and chimpanzee A3C have alanine at position 144. So we made two more mutants of rhesus A3C. The single mutant has alanine at position 144 and the triple mutant has arginine at position 44 and 45 and alanine at position 144. We did the single cycle infectivity assay and found that the triple mutant of rhesus A3C has almost same restrictive activity against HIV-1 as human S188I. Again, the expression level of all the construct was similar and all of them encapsulated in virion at similar level. We purified the proteins and did size exclusion chromatography. As you can see, the S144A triple mutant existed predominantly as a dimer. We did the in vitro deamination assay and the specific activity of the S144A triple mutant was approximately 2-fold more than the wild-type rhesus A3C. To conclude, APOBEC3C has evolved under positive selection in primates, suggesting it played a role in blocking primate lentiviruses. Old world monkey rhesus A3C is not active against HIV-1 but gained activity in hominid lineage. The key amino acids required to increase activity of rhesus A3C are at position 44, 45, and 144. Similar to human and chimp, A3C specific deamination activity is correlated with its ability to dimerize. In the end, I would like to thank my supervisor, Dr. Linda Chalico, Chalico Lab members, and our collaborators at University of Texas at Dallas, University of North Texas, and Fred Hutchinson Cancer Research Center. I will also like to thank my funding agencies. And I will also like to thank you all for your patience and attention. And I am ready to take any questions.